Great. So we're now going to move on to the panel session. And I would like to introduce the monitor for this session, Chris Wyatt. Chris was the founding president and CEO of the Caring Colorado Foundation, serving from 2000 until his retirement in 2020. Previously, the executive director of Colorado's Tri-County Health Department, Dr. Wyatt has extensive experience with health departments, including collaborating with communities dealing with environmental contamination. He was the chief of environmental chemistry of the environmental chemistry section of the Illinois Department of Public Health, and actually done a significant amount of work advising around drinking water and other environmental standards. Chris, for that, I'd like to turn things over to you.
Thanks, Ned. So our first panel session today, the topic, current knowledge about the contribution of PFAS exposure sources to human exposure. And our speaker is going to be Elsie Sunderland. Dr. Sunderland is the Gordon McKay Professor of Environmental Chemistry at the School of Engineering and Applied Science in the Department of Environmental Health at the Harvard School of Public Health. She's also an affiliated faculty member of the Department of Earth and Planetary Sciences. She's a faculty associate in the Harvard University Center for Environment and the Harvard Center for Risk Analysis. So without further ado, I'll introduce Elsie Sunderland. She'll make a presentation. We'll then introduce our responsive panel. We'll have just a few minutes for questions, then introduce our panel. They'll provide a brief reflection on Dr. Sunderland's presentation, and then we'll have lots of time for questions from the committee. Dr. Sunderland.
Okay, thank you very much. Are these slides sharing?
Yes.
Okay, great.
You see them in like the mode where I can see your other slides at the side. There you go.
Good, great. Okay, well, thank you to the committee for inviting me to speak to you today. I've been charged with talking a little bit about our current understanding of sources of human exposure to PFAS and what we can say at this time in terms of quantitatively attributing the major sources for different demographic groups and different populations and communities across the country. So I wanted to start with this slide, which gets us thinking about some of the different potential sources for PFAS. And as soon as we start to think about exposure sources, we start to recognize the tremendous diversity of sources. I wanted to point out that there are two different timescales of chemical production integrated in what humans receive, and this can be important when thinking about the sources. So we have chemical production of different compounds and a direct pathway through consumer products, so outdoor gear, food packaging, personal care products, and cosmetics directly to humans. And then through our waste infrastructure, through use of large sources like aqueous film forming foams, we have an environmental distribution pathway. So the PFAS are released into the environment, they contaminate drinking water, they can get into our agricultural foods, they can accumulate in seafood, and they can persist in the environment for a very long time. So humans can receive through their diet and through their use of products an integrated signature of both contemporary chemical production, but also a signature of what we now call legacy PFAS production that still remains in the environment. And so thinking about those timescales, when you look at the composition of PFAS in human blood or serum, that does provide some clues as to some of the sources, and we can come back to that as I talk about the different sources today. So unfortunately, my conclusion is already on this slide, and our state of knowledge in terms of understanding and being able to quantify the relative significance of different human exposure sources right now is quite limited for the general population. What we do understand is that for contaminated communities, so on the right in the slide, we understand that drinking water, when you have high concentrations of PFAS in drinking water, this is the dominant exposure source. As soon as we shift away from those contaminated communities, and in some communities where you may have an air emission source, and this is being increasingly recognized, then you can have several different predominant sources. So that if you have an air emission source, you have a pathway then potentially into, again, that agricultural pathway, so into food and other sources. For the general population, in the United States, we really have limited data that allow us to attribute the relative importance of different sources. What we do have is, clues as to what's important, and I'm going to walk through some of that now. And I've already mentioned the sort of three bins I want to go through today. The first is our main understanding of drinking water and where we are in terms of for both contaminated communities and for the general population. The next is diet or food sources. And the third bin, shall we say, will be consumer products. And I'm including or lumping into that dust as a vector of exposure for some of those products and in the indoor environment. And just a footnote on this slide, what we do have, so we have limited information attributing the relative significance in the general population of these different sources, but we do have pretty good biomonitoring data. So similar to the European studies, and I'm citing some data from the European Food Safety Authority here, we know that young children are some of the most highly exposed populations, and that's particularly concerning, and that early in life maternal exposures are also an important exposure source. So if I go through now these three different bins of exposure sources, so starting with drinking water and then going to diet and consumer products, we can talk about our state of understanding in these three areas. And I've already mentioned that for contaminated communities or next to contaminated sites, that's where we have the best understanding of the main exposure pathway, i.e. it is drinking water. And that drinking water needs to be the focus of remediation efforts or interventions. What we're seeing over time, and this is a little bit out of date, and so this continues, and there's some updated graphics that I think you've seen in previous presentations, is that the number of contaminated sites is expanding dramatically. So our understanding of where that is a concern is still growing all the time. So there are many, many sites, and we're discovering more every day. If we switch now to thinking about, well, what about the general population? This is a nice study relatively recently from the Environmental Working Group, looking at the number of individuals exposed to tap water PFAS, and in this case, they were just considering PFOA and PFAS concentrations above different thresholds. So if we pick a threshold around 10 nanograms per liter, which is where many of the states are starting to fall with their drinking water MCLs or maximum contaminant levels, we see that there are between 18 to 80 million US individuals who have PFAS in their tap water that exceed that level. So this obviously has sparked a lot of concern for a number of people. And I think the next question we want to ask is, well, in that range, so between 1 and 10 nanograms per liter, how important is that drinking water source relative to some of these other sources that we know exist? And you can do that kind of an evaluation by combining biomonitoring data with source data. And so I'm showing you an example of a study like this that's come from, it was led by my doctoral student, Cindy Hugh, a couple of years ago, working with a cohort based in the Harvard School of Public Health, the Nurses' Health Study. And this study was actually an example where tap water samples were obtained in combination with blood samples at the time. So we have an opportunity to evaluate the contribution of drinking water to the actual measured serum concentrations. And we're talking about in this cohort, so this is a national cohort of US women that has characteristics very similar, so that in terms of demographic attributes, it's very similar to the NHANES women cohort. So it's a US general population of women. You can see the tap water concentrations. I'm going to advance one slide, are in this range that I was showing you for the environmental working group, 10 nanograms per liter. In both cases, so when I advanced the slide, I removed a high leverage, so one highly exposed individual. And in both cases where so looking at PFAA and the C9 carboxylate, PFNA, for individuals who consume greater than 8 cups of tap water per day, even at these relatively low levels, the concentrations in the tap water are statistically significantly associated with levels in their serum. So drinking water is still important at these levels, but they contribute on the order of, so we propagate the drinking water exposure through to the serum concentrations using toxicokinetic modeling. They account for, and that's what this RSC number is, so the relative source contribution is between 2 to 34% of their overall exposure. So what does that mean? I think it means that water is still an important vector, but if you invert this argument, it also means that many other sources are important when we start to think about the general population. I wanted to add this next slide as a note to all of you. I know it's slightly beyond your charge as a committee, and you've been tasked with looking at a few very specific PFAS, but the chemical family is extremely large. It contains thousands of compounds. And one of the things my research group has been doing, and other groups have been doing this as well, is trying to think about, well, how, what is the total burden of organofluorine out there, and how much are we actually accounting for when we look at these few targeted PFAS? A variety of the burden of a good portion of the burden of that organofluorine in the environment consists of what we call PFAS precursors that degrade into some of the compounds that the committee has been charged with looking at. So I'm not sure if that falls within your mandate, but I think it's important because when we look at so here going back, if we look on the right, so the slide on the left is pointing out, I didn't show you the data from the paper, but I just wanted to highlight that we find large quantities of unidentified organofluorine in watersheds around Cape Cod, where we've been looking. In the tap water samples that we considered in the drinking water study that I showed you previously, we compared tap water levels between 1989 and 1990 and resampled the same public water supply in 2016. And we see a large jump in that unexplained organofluorine burden in the environment. So this is one of the most critical issues, I think, that is still outstanding in this field, and something that we really need to get a handle on in terms of exposure. I'd like to transition now to thinking about dietary exposure sources for PFAS. And I started by saying, or by quoting the European Food Safety Authority assessment. And they're pointing out in this assessment that many sources of dietary sources contain detectable burdens of different PFAS. The story is complex because it varies by the PFAS being considered. It varies by the demographic group being considered, but diet is thought to be the prevalent source of PFAS exposure for the general European population. And that really contrasts what we have in terms of data or information for the US population right now. And I'll speak to this point a little bit more. So in the European study, then, if we're thinking about the example of PFAS, for example, in the adult population, then seafood and other meat and egg products end up being a dominant source of exposure. And we see this picture or evidence for a similar picture in the US population when we look to the epidemiological studies. So we're looking at serum concentrations of different PFAS and the association with different dietary indicators. So you see, and this is just one example from our work, you can see this in many different epidemiological studies when you look for correlates with the measured burden of PFAS in blood. Then you see, and in this example, I'm showing you the C10 carboxylate. You see correlations with the amount of seafood consumed. In this case, it's N. Haynes 2005-2006, and the number of shellfish meals. But seafood, often jumps out, and that's not surprising because some of these long-chain PFAS, so PFAS with more carbons attached to them that tend to be produced more abundantly in previous decades, accumulate in food webs. And we've actually, in our own work, proposed that actually, if you have biomonitoring data for an individual and you're trying to understand what are dominant sources of exposure, and you see some of these long chain PFAS, so things like the C10 or the C11 carboxylates, that can be a clue that that individual may be exposed to some degree through their seafood consumption. So that's something that you can also use in kind of a diagnostic capacity. If we move away from seafood and we start to think about other foods, so crops, vegetables, different meats, and milk and eggs, there are examples in the US population where we know that this is a concern. And I've just highlighted in this slide the example from Maine, where this has been an issue that was discovered where an industrial release of PFAS or industrial waste was basically mixed with municipal waste in a biosolids waste stream that was spread on agricultural fields. The PFAS in the agricultural fields then accumulated in the different crops, which then accumulated in the various animals, including the cows and the milk, and of course then the farmer himself. And so the state of Maine is quite advanced in thinking about this and in thinking about this agronomic exposure pathway. There are other examples in the literature where this has been quantified and thought about. I don't think, again, we have a good systematic understanding of the extent of this problem across the population and its implications for exposures of the general population through food. We have these anecdotal examples instead. So this is something I think where we need some work, but we have clues that it's important again. The next pathway that I wanted to talk about whereby PFAS can get into food is through food packaging itself. And there's a lot of data on this. Tom Webster, who's on our panel, sent me a European study assessment about 5 minutes before we started, showing a lot of data from Europe. Again, we have data for the US and North America in general from some nice work by Laurel Shader, who's also on our panel. Where we see the prevalence of organofluorine and targeted PFAS in these different food packaging products is quite abundant. We've done some work in my lab looking at this. This is the panel on the right, where we're looking at contrasting again those targeted PFAS with total organofluorine measurements. And, if you think back to what I said when we began, about the time scales of chemical production working their way into exposures, we're seeing a lot of very new PFAS showing up in these consumer products that perhaps we're not measuring directly at this time. This is because of we have these data on the prevalence of PFAS at very high levels in food packaging. What we don't have is the direct link between that and the packaging as an exposure vector or its relative significance. In my lab, we have been doing research at an AFFF contaminated site on Cape Cod, and we were doing work on compostables, looking at the PFAS burden in compostables. And after a 15-minute methanol leach or a DI water leach with that compostable, we found concentrations in solution that exceeded the highest concentration at that AFFF contaminated site. So again, there are clues that this is a problem, but the mapping out the entire pathway all the way to human exposure hasn't been done yet in a quantitative way. We have these clues, as I showed you for seafood again, in terms of associations between serum concentrations and individual interactions with packaged food. So here I'm showing you an example from a cohort in Vancouver, Canada, with the C6 sulfonate and PFAS. And you can see that the use of, or the increasing use of prepackaged foods or food packaging is associated with higher serum concentrations. So I guess all of this discussion on food brings me to what we've heard very recently from FDA, which is a very definitive statement about the safety of the US food supply. And I guess I wanted to spend just a moment with the committee contemplating this statement and perhaps, what should be the charge when we evaluate the significance of dietary exposures for PFAS in the US general population. So the findings of almost no detectable PFAS in food are somewhat surprising. So only 4 samples out of 300 tested had detectable levels of PFAS in the products tested so far. And that really contrasts the great abundance of data that has been coming out from the European Food Safety Authority, which has reported, as I mentioned, PFAS, very high levels of PFAS in some cases in many different foods. And they've established a tolerable weekly intake recently for 4 different PFAS, and I'm showing you the number in this slide. And if we look at the detection limits reported by FDA at this time, you can do a back of the envelope calculation that says even at the detection limit, and given the magnitudes of certain foods and certain meats consumed by US individuals, exposures could easily exceed that threshold set by EFSA. So I personally am not comfortable dismissing this as something that's safe at this time. I also think that the charge question of sort of what is the contribution of diet to PFAS exposure in the US general population has not necessarily been analyzed or answered by in the selection of samples or with the representativeness of the food samples that have been measured so far. So I hope for more in the future with that. I'm also surprised by the low incidence of detection, because if you look, for example, even just something like seafood, and you look at previous FDA work, and I've cited a paper from 2013, where the detection limits for that study were about an order of magnitude higher than the recent work. And they still found detectable concentrations in randomly selected grocery store seafood samples in 11 of 46 samples. So I think this still warrants additional consideration and discussion. I will wrap up here by talking about personal care products and what we know. And it's a similar story to many of the themes that I've touched on before. There was a paper recently released, spearheaded by the Green Science and Policy Institute, showing that there's a very high abundance of PFAS in many different cosmetic products in North America, and often those products aren't labeled. And that's a similar story to what we've been finding or what we'd seen in the literature for Europe. And the study that I'm citing here is a paper that looked at the Swedish market, and that the issue with these studies in terms of taking an abundance detection for PFAS and translating that into a human exposure or a quantitative exposure estimate is that we really don't understand a dermal absorption of different PFAS at this time, and there's very little data on that. And so this one example used a single study for PFAA and the ranges reported for absorption efficiencies in that study to say, hey, well, given those ranges, the levels of PFAS that we're seeing in various cosmetics in the Swedish market, which are similar to the North American market, could be, something small, or it could be something that rivals dietary exposure, or even exceeds dietary exposure. So again, it's an area where we need more work. The other thing that we do know is that there's a transfer pathway from things like furniture and products and high levels of PFAS into dust. So when we look at measured concentrations in dust, which many studies have done, we see very high levels of PFAS, and we need more work thinking again about how relevant that is in terms of body burden of PFAS and how that could be translated into exposure reductions. So in this example that I'm showing you is a study of 15 fire stations in Massachusetts looking at dust in different rooms, showing that there are different levels, and it turns out that the turnout gear was actually a substantial source of PFAS and elevated PFAS and dust in some rooms. So if you try to bring the numbers from various ad hoc studies together, which we've done in a few different synthesis efforts with the community over the past few years, the most recent one I'm citing, this De Silva et al. 2021 paper, which was sponsored by a CTAC PFAS workshop, then we see a summary of what I've been saying to you basically. So there's a table in this paper where we've synthesized the different studies, and you can see the attribution from different sources. But our general feeling is that inhalation or direct inhalation is likely relatively small. Dermal uptake, we really don't have a good handle on at this time. And there are these 3 different bins of diet, dust, and water, which could account in the general population for anywhere from a very small fraction of the exposure to basically all of the exposure. And I guess at this time, what I would say is we can use the composition of the PFAS and our knowledge of that to try to tease out or do some investigative work on what could be the dominant sources for different populations. But we need more work pairing, so more systematic exposure studies pairing biomonitoring data with exposure information, and we really haven't done this very systematically yet, and there haven't been resources dedicated to this. So that would be the next advance. And I think I'm out of time, so I'm going to stop there. And thank you for listening. And I'll hand it back to the panel for discussion.
Thank you, Dr. Sunderland. Really interesting work on occurrence. It's something that it's clear we need more information. We have a few minutes for some questions before we go to the panel, and while the committee members are thinking of those questions, I'd like to ask you, talk a little bit about your sense of the feasible exposure reduction methods. That, I mean, it's clear these compounds are ubiquitous, and some reduction may be easier than others. For instance, occupational exposure to firefighters, we've heard some about. Talk more about your other thoughts about how exposure reduction might take place to reduce that exposure and reduce health outcomes.
Yeah, well, that's a great question. Thank you. I think, there's sort of, we don't have all the numbers to attribute exposures for different demographic groups right now, but we know that there are a number of prevalent sources. So, things like pizza boxes and microwave popcorn, products that contain high concentrations of PFAS, all of those things, those are a choice, and those can be, you can steer away from those. I know there's some discussion about how transparently information on PFAS content of products is advertised. I think there are a number of groups right now working to make that information more transparent, which can be helpful. And I was, if drinking water is the exposure vector, then that is something that can be removed as an exposure pathway very quickly. So there's a nice paper actually from the North Carolina group showing the effectiveness of different point of use water filters in homes and other places. So I think those kinds of actions can be taken. I think the Environmental Working Group publishes a very nice list of safe cosmetics and tries to stay on top of the chemical burden in those kinds of products. And so those kinds of interventions are easy as we work on this problem as a whole.
Good, thanks. Jane Hoppin, do you have a question?
Yeah, thanks, Elsie. It was a great talk. So I'm particularly interested in kind of how we do the comparison between environmental and biological samples, because, the biological samples the PFAS could have a half-life of 5 years, let's say. And the environmental samples measure today. And so what are we missing? Like, because the time horizon for the two measures is different. What are your thoughts on that?
So you mean in terms of, like, if we're looking at a snapshot of human serum, and then we have some contemporary exposure vector, but that we're basically assuming that that's been constant over time, or we need to reconstruct the historical changes in that. Is that what you're asking, Jane? Yeah.
So if we assume, I'll just use like half-life of a chemical is 5 years, and 5 half-lives is complete elimination. What is in someone's blood could be 25 years ago, and we're measuring today. I don't know. I'm kind of curious. I've been thinking about this, so I'm curious about what your thoughts on it. Like, how much error are we introducing in trying to use these exposure measures of today to look at these markers of today and up to 25 years ago? How do you think there's error in that question?
Yeah, I mean, obviously there is error, and if there is historical data available on shifts in exposure, then I think we can work with that. I guess my experience at some of these contaminated sites is that the concentrations have been, there was a lot of historical use of AFFF at a lot of sites, and then the concentrations in the groundwater around those areas, unless there's a remediation intervention, seem to be relatively stable. So that, for say you're reconstructing water as an exposure vector, I think a first cut that assumes steady state is okay. If there's an intervention in an unknown time period, then that can be accounted for. When we try to do more complex work associated with that in our environmental modeling, then we actually look at histories of chemical production and try to incorporate that into the exposure signature for people. But it's a challenging and interesting question. I think in the cases where we have time series data, you definitely see changes. So a lot of work that we've done is just looking at the response to that elimination of POSF, so PFAS and its precursors, and what that did to exposures over different timescales. Because we actually, through PFAS and PFOA, have a case control study of what happens when you eliminate and how quickly do you see these changes. So we can perhaps apply that knowledge forward to some of these other compounds.